So before we move on, I would like to, or in order to move on, I would like to introduce our next two speakers, Dr. Alex Nord and Dr. Martine Therrien. Dr. Martine Therrien is an assistant professor of molecular and cellular biology and core faculty at the Center for Neuroscience. She's a campus scholar and the Neurofest 10th anniversary chair. Her cutting-edge research uses induced pluripotent stem cells to understand how genetic variants affect the initiation and progression of neurodegenerative disorders and rapidly translate her findings to humans. An important focus of her work is how the immune system and immune cells, called microglia, can be used to track and modulate disease progression. Today, she will discuss the development of platforms to interrogate the effects of genetic and brain environment on microglia states and functions in Alzheimer's disease. So she will talk at 1:55 p.m. Our first speaker now is Dr. Alex Nord, who's Professor of Neurobiology, Physiology and Behavior and Psychiatry and Behavioral Sciences. He's a core faculty member at the Center for Neuroscience and an affiliate faculty at both the Mind Institute and the Genome Center. Dr. Nord's research explores gene regulatory circuits and chromatin dynamics in the brain and how these features contribute to brain development, evolution, and function. An important focus of his current work is understanding how mutations that cause disorders like autism and epilepsy impact the brain. Early in his career, Dr. Nord contributed to the development of methods to bring DNA sequencing into the clinic, focusing on familial breast and ovarian cancer, and on autism and schizophrenia in his Ph.D. research. For his postdoctoral work, he moved from searching for mutations to working to understand the genetics of brain development. His work is rooted in basic and translational science, with the ultimate goal of understanding the biological components of human diseases and disorders of the brain and improving clinical care of afflicted individuals. To this end, he performs both experimental work and computational analysis to reveal functions of primary DNA sequence, epigenomic modifications, and chromatin structure, and uses cellular and animal models to understand the molecular roots of brain disorders. Today, he will talk to you about new genetics technologies that reveal the inner lives of neurons. Please join me in welcoming Dr. Nord.
Thank you. Diasnu, is that volume okay back there? Well, getting help set up here. I'll give a quick introduction of myself. I wasn't a neuroscientist until I came to UC Davis as a faculty member 10 years ago. I was trained in human genetics, and in my PhD, I worked with clinicians to try to find better ways to find the genetic mutations driving cancer and brain disorders. So I worked in familial breast and ovarian cancer, and in autism and schizophrenia. When I finished my PhD, I decided that I wanted to kind of dive deeper into what mutations were doing, and so I did a postdoc working on brain development and using new genetic tools to understand how our genomes control our brains. And that eventually led me here, because UC Davis was looking for a neuroscientist who had a strong genetic background, and I could at least fill the genetic background box. Since then, I've had the pleasure of working with my colleagues at the Center for Neuroscience and at UC Davis, have taught me a huge amount about neuroscience and helped me to build a true brain-oriented research program. So let me make sure everything's working here. Do I have to go? All right. Okay, so the brain, to me, I start with the nucleus of the cells within the brain, and I ask questions driven from a goal to understand the genetics of the brain. So there's an intersection of two fields here. The kind of overarching goal of what I do and what geneticists like I am working on is trying to understand how our DNA contains the information, the blueprint of our brain, development, function, how the cell types in our brain develop, where they're born, how they develop, and the connections they form, and how the genetics determines that. Then from a disease perspective, how mutations alter how cells in the brain, I'm using neurons here, but we also care about the glia, how they change how those neurons develop or behave and lead to the pathology that underlies disorders. And my work's mostly focusing on the early onset brain disorders, kind of severe neurodevelopmental disorders, autism, and epilepsy. And then finally, what I'll talk most about today is how we can harness some of these new genetic advances to build tools to help further neuroscience research and eventually treat brain disorders. Normally, I like to tell kind of a more detailed story about some part of our research, but today I got asked to cover a decade of research and to talk about tools that have helped further our understanding of the brain. So instead of giving you kind of in-depth ideas, I'm going to go through some of these tools and talk in a quick vignette about how our lab is using them. So the first, maybe the most famous, the CRISPR-Cas system. Raise your hand if you've heard of CRISPR-Cas before. I won't ask you to raise your hand if you can explain to me how it works. That's a challenge for the undergraduates I'll be teaching next quarter, even after my lecture on CRISPR-Cas. This was discovered one among many routes. Many of the discoveries you hear about are not just one person discovering, but are a team or many teams. And one of the fun sides of this is it was a yogurt biologist that made one of the key initial discoveries that led to our understanding of CRISPR. I don't have time to go into that today. I'm going to tell you a little bit about how this tool has been repurposed for biotechnology and is used in the brain. So in this cartoon here, what you're seeing is a stretch of DNA. So we'll have a couple cartoons of DNA here. It's a double-stranded molecules with base pairs that connect. And importantly, the information of DNA is contained on each of the strands together so that it can be copied or it can be read. And the way the CRISPR-Cas system works is that there are two components. One is a protein called the Cas9 endonuclease. There are different forms of this. I'll just talk about this as Cas. It is a protein that's capable of reading along DNA and cutting or forming a double-stranded break in the DNA. And it's an initial first life as a bacterial immune protein. It was used to attack bacteriophages, or the viruses that attacked bacteria. And in that initial research to try to understand what would make the yogurt bacteria resistant to such viruses. But we've repurposed this to now enable us to target DNA in a programmable way. And it's the second component of the system that it makes it so powerful. And that is the RNA that is loaded by the Cas protein and used to read along the genome. And as I said, DNA is double-stranded and there's same information on either strand. RNA is like a single-stranded DNA molecule in that it can form the same kind of interactions. And so through these two components, we can give the Cas protein any synthetic guide RNA, an RNA that guides it to where its target is, and then it will make a change, or in this simple form, it'll make a cut. We can thus use this to target anywhere in our genomes and introduce a change, whether those are random mutations, which we use, for example, in disease modeling, or to introduce specific versions of genes in a research context or in the future in a treatment context. So this system is kind of one of the, thought to be one of the transformative technologies of the century, has allowed us the access to basically edit and engineer in a precise mechanic our DNA. And as just an example of how we're using it or how it's useful in my world, we're interested in severe mutations or mutations that cause severe neurodevelopmental disorders. For example, we study a gene called CHD8 that humans that carry mutations of this gene have severe cognitive and social deficits, as well as brain overgrowth. And this is an image of an MRI from a patient that was seen at the Mines Institute that carried a CHD8 mutation, where the degree of red here is the degree of overgrowth of the cortex. We were interested in studying this gene, and we used the CRISPR-Cas system to engineer a mouse to carry a mutation that allowed us to study it in mice, what was changing in the brain. And I'll show some pictures of people in my lab and our collaborators. Cesar Canales is a research faculty in my lab, and Stefano Lozano is a grad student. Both of them are currently involved in trying to understand what the differences in the brain caused by this mutation. But just as a quick vignette of the power of this, the mouse model that we generated shows the same type of overgrowth in the brain you can see in the mouse MRI now, where the pink color is where it's larger than its non-mutant litter mates. So we can use this tool to rapidly engineer these systems, whether it's in a mouse, in a cell line, or in a human organoid. Moving on, we're going to use the same kind of framework to manipulate other steps of genetics. So if you can think back to an intergenetics class or where you've encountered this before, we have a central dogma of genetics where we have DNA, which contains the genetic information, transcribed to RNA, which is an intermediate messenger molecule, which is then read out through translation by ribosomes that build the proteins. So the DNA is the instruction manual, and RNA is an intermediate message that allows it to be more efficient and more flexible, and then the proteins are what make cells actually work. Using the same discovery of this CRISPR-Cas system, one of the first ways to manipulate this system is we can take the Cas9 protein, and instead of using the endogenous form, which can cut the DNA, we've made a new synthetic form which can activate transcription or turn on production of RNA, or on the flip side, inhibit it. So now we can control, basically we have switches to control which genes are turned on and off using the same system, and it's the same programmable nature by our RNA so that we can use this to target basically any gene in the genome and alter how much of it is produced in any cells. We can also alter this second step, translation, using something that was discovered in the late 1990s but took a while to mature, and that's that we can use RNA to interfere with translation. So in this case, an RNA is produced from a gene, and then we bring in what's called an antisense oligonucleotide. Antisense meaning it's the opposite of its target RNA, and oligonucleotide is just a short stretch of bases. When that interacts with its target RNA, it can inhibit translation or cause the protein to no longer be made, or if we use it in a different way, we can turn up translation to cause more of the protein to be made. So using these tools, we can basically engineer first the genetic code using the Cas9 system, then we can turn on or off or control how cells use the DNA, and finally control how proteins are manipulated. And as an example of this in the same neurodevelopmental disorder context, we're interested in these genes that when mutated cause these severe phenotypes. And in work we've done with a mouse model, and this is with a colleague, Jill Silverman, School of Medicine and the Mine of Psychiatry, and a postdoc that was with my lab, Jess Haag, is to try to understand, can we turn back up the amount of genes that are necessary for normal function in the brain for some of these mutations? So this is a gene called SCN1A. It's a channel that neurons make that is important in neuron firing. And individuals who have a mutation in one copy of this gene have a severe form of epilepsy called Dravet syndrome. So this is the same picture I've been showing you, but this is actually a picture of the mutant. In a healthy mouse, the SCN1A makes this NaV1.1 protein, which is shown in green here. And so the idea is here is if we can take these tools to alter how much cells are making, we may be able to rescue these phenotypes for treatment. So in this case, we use this CRISPR-A system, bring a guide RNA that targets the SCN1A gene, brings the Cas9 protein to the SCN1A gene in the genome. And then what our goal is and what we've done in cells is we can increase the expression of that gene back up to how much is in a normal mouse. And now we can go test and ask, can we rescue phenotypes? And this is an exciting treatment avenue for some of these disorders. Hopping forward to the next technology. In addition to the ability to do engineering and to change the kind of molecular makeup of cells, we also have new abilities to measure what cells are and what they make at the level of single cells across thousands of cells at once. So these technologies are premised on our ability to take a complex tissue like the brain, whereas, for example, when I started here, what we were forced to do is if we wanted to study this and ask what RNA is made, we'd grind up the brain. Sometimes we'll show the Lego, sometimes show the blender. Basically, we lose all the information about individual cells. So using these new technologies, we can retain the information about the cells that we're studying and not only profile individual cell types, but actual individual cells within a complex tissue. To run quickly how this works, we can take a brain, isolate individual cells or the nuclei from cells, and then encase them in droplets. And inside that droplet is the chemistry needed to generate all the RNA from that cell, all in one droplet. Also in that droplet are barcodes that are added to each RNA molecule, so that when we can sequence, we can track where those barcodes came from, and that allows us to track back to which cells they came from. This allows us to measure every RNA in every individual cell in a sample. And this cartoon here showing how this works, each dot here would be the measurements from a single cell, And they're clustered together in this plot based on the similarity of the RNAs that are produced. And then based on what those are, we can go back and figure out which those cell types are. So shown in these pictures here, we have neurons, oligodendrocytes in orange, microglia in purple, astrocytes in blue. We can look at each of these populations in one study. We can identify the cell types, and then we can go and measure the changes in individual cell types or even individual cells to learn about what's happening in this complex tissue. Example of how we're using this in my lab. We use this for many studies now. We're able to get this kind of refined perspective of what's happening. One of the fun new ones is we've engaged in a collaboration with David Olson here to try to understand which cells in the brain are responding to psychedelics in a mouse brain when given a dosage. It's work that in my lab is led by a grad student, Ethan Fenton, who has a poster on this here. So in the experiments here, we administer the drug. We wait an hour, which is just enough time for cells to start responding, start turning on new expression of genes that are in response to that psychedelic. And then we perform our cell isolation and our single-cell RNA-seq analysis to identify all the different cell types in the mouse cortex here. Each color here is a different cell type. And then we can ask which of these cells are showing a response to the psychedelic. So Ethan built an algorithm here to pick up cells that appear to be responding to the drug, and then to look at specific neuronal populations. What he found is that there's this one population here that are excitatory neurons in the brain that seem to be these primary neurons that are activated by this psychedelic drug. This gives us information about targets, about circuitry, and then allows us to also ask questions. What is the downstream response? What might be the difference between different drug types and which cells they reach or what pathways they activate? This was just simply not possible without having the single cell perspective, because as you can see, there are cells across all these different clusters of cells that do respond, but it's only this one cluster in the middle here that has kind of a more uniform response. Okay, I'm gonna give you one more before we sum up the technologies. The last three I talked about were ways to either manipulate or observe the central dogma of DNA, RNA, or protein. I'm going to talk about one additional technology that's use of viruses to access neurons in the brain. Scientists have used viruses for a while to be able to observe neurons in the brain, to be able to do things like track connections. The work I'm going to talk about today comes from a specific class of viruses called adeno-associated viruses. These are special because they're capable of infecting many cell types, including neurons in a brain. And they're relatively non-immunogenic. So these are an exciting class of viruses, for example, in gene therapy, because they have a reduced response. So cells don't change much when they're infected with these viruses. The way the AAV, as I'll say, adeno-associated viruses work is they form a capsid or a capsule that encloses a material. And like any virus, they access cells through gaining access into the cell, in case through some specific receptor. Once they're inside the cell, they then deposit their payload of whatever is inside the capsid. It's a genetic payload, so it's DNA, and deposit it inside the cell, so the cell now carries that new DNA that we've introduced. These AAVs cannot package themselves, so they're not infectious. They don't continue to infect, but they can be used across a different variety of organisms, of cell types. And in the case that we're using, how this is genetic, is our payload is DNA that allows for expression of certain genes. For example, in a gene therapy context, we might provide a healthy gene to a cell. In a mouse, we can take the AAV, package our genetic payload, inject it into a mouse brain, and then read out what's happening. In this case, this is an imaged mouse brain showing this GFP or green fluorescent protein for cells that were infected. And an example of how we're using this, or what the power of this technology is in combination, the capsid and the genetic payload allows us to target with great precision specific cell types in the brain, and then we can build tools to manipulate these cells. So here, in this example, I'm talking about work with Dr. Fioravante who introduced me, where we're trying to access and manipulate specific cell types in the cerebellum. This is work with our lab that I'll show from a graduate student, Corey Articani. So in the cerebellum, there are a number of cell types that form these known circuits. You'll have to ask Dr. Fioravante about those. And our goal was to be able to access different cell types that are involved in different steps of this. So we developed genetic payloads that would enable the virus to only be effective in certain different cell types. So in this case, showing us the two cell types. These are targeting Purkinje cells in the cerebellum, where this virus is specific to this molecular layer interneurons. You can see the differences in the patterns. The viruses infect both cells the same, but they're only turned on based on the genetic payload. Okay. So where I'll leave you with some of these technologies is that not only is this enabling basic research, but it's also enabling changes that take a few more years but get to the clinic for treatment and diagnosis of of brain disorders. Skip ahead a little bit here. Did we start on time? So I've talked about 4 of these technologies. We can edit DNA with CRISPR. We can alter neuronal or cellular protein production by changing the amount of RNA or protein that's produced by these cells. And we can read the cellular characteristics of specific cells to learn which cells do things or are responsive to things. For example, which cells respond to psychedelic drugs, are impacted by patient mutations, or have evolutionary differences between mice and humans. Then we can manipulate these cells using these engineered adeno-associated viruses, or AAVs. My lab, most of our experiments now combine across these kind of technologies to identify which cells are impacted and then manipulate those cells with an AAV, or for example, rescue the cells that may be causing pathology. I don't have time to tell you about some of these treatments today, but I just want to mention that these, in addition to being powerful for research, are also now changing diagnosis and treatment. For example, antisense oligos targeting SCN1A in December 2024 were recognized by the FDA as a breakthrough therapy and are now beginning to be used in patients to provide a cure for the intractable epilepsy of Dravet syndrome. The first AAV gene therapy in the brain was approved by the FDA last November, and we're on the verge of seeing some of these other technologies be used in the clinic, such as CRISPR gene editing, which is already used in blood phenotypes but not yet in the brain, and use of these single cell tools, for example, for tumor profiling. The last thing I want to leave you with is just the note that none of this is possible without basic research funding. Sometimes there's some confusion about where does the money come to support this kind of research. Basic science is a term that captures work that's done that doesn't have obvious utility or financial value. It's not that it won't or that you can't see why, but it's too far from the market to be supported by industry. The findings I showed today grew out of this kind of basic research that's only possible with the kind of research investment that we get in this. There is really no reason for private sector to fund these works yet. There's not close enough to know what to do. So we depend on the funding for basic research from philanthropy and from the federal government. Ask us, if you're here today, like the first question, ask us how these research dollars make it possible for us to do this research that eventually will drive then these changes, that breakthroughs in medical technologies, as well as providing financial benefit, jobs, economic improvement. All right, and then I didn't get to say much about the work we do in depth today, but I have a wonderful lab of PhD students, a few scientists, and amazing undergraduate student volunteers from UC Davis. It's one of our the most valuable resources here is running a lab is how many students we have that are excited to pitch in and help. So, for listening.
Questions for Dr. Nord?
Thank you. I'm wondering how close do you think we are to approving genetic therapy for Huntington disease, which my understanding is similarly caused by a genetic disorder or inherited in the HTT gene, like Dravet, like you pointed out in 2024.
Yeah, it's not my area. I know it's a primary target because of its prevalence and its known genetic causes, but I would turn that over to someone else. Because I'm not sure where that particular pathway or pipeline is.
You talked in great deal about using mice brains to do the research. I'm just wondering how much of this is being applied to either living or deceased human brains.
In research context, all these tools that I talked about today have been applied to both material from resections, from surgical resections, from living human brain, or from deceased brain. If you're talking about a clinical utility, then there's a lot more steps, and that's why there's some space between what we do in mice you start to see in the clinic. But as I said, the AAV gene therapy was recently approved in humans as a treatment, as were the ASO therapy, and those are both delivered as a kind of not a full-on cure, but a much more effective treatment than was out there that's based on symptoms. So, yeah, it's not a limitation to mice.
Last question.
Yes, all the technologies you talked about involves targeting specific gene mutations. Are there gene mutations associated with age-related chronic diseases for neurodegenerative diseases that have been identified that are targets for gene therapy?
Yes, it's not my area, so I don't use those as examples. I also was focusing on situations where we call it monogenic, where one mutation causes a disease, and that's not necessarily the case for a lot of disorders, especially some of the neurodegenerative disorders. But yeah, for example, the ApoE allele in Alzheimer's, there's attempts to do this kind of engineering with.
But the problem is heritability. It's not genetic.
So there's two answers. One is most human diseases have genetic components. And the second is that the environmental components of human disease also work through some of the same pathways. Yeah.
But majority of the components of phenotypic variation, most of it is genotype by environment, not genetic, for most of these complex genetic diseases.
Yeah, it's a fun conversation. I'm happy to continue having. Disease is yes, there's genetics, there's environmental aspects. Some of the things that we can learn from genetics, the pathology follows the same molecular pathways in the brain for environmental aspects as for genetic aspects. So sometimes genetics can give us information about the environmental causes. But yeah, great discussion to continue.
Thank you so much.